Hello, welcome, and this video is going to be all about mitosis and the different phases of it. So there's 4 phases total, and how you can remember this is with this little slogan, you might say, PMAT. And each letter stands for a different phase of mitosis. So Prophase, Metaphase, Anaphase, and Telophase. And we will go into depth in them in just a second. Alright, so the first one is prophase, and how you can remember this is that it starts with a P, and the P stands for preparing. So the cell prepares itself for division. So the chromosomes become more organized. Before, they were chromatins. Chromatins is DNA tangled how it is usually during interphase, when the cell just does its normal metabolic functions. But as it gets ready to divide, they become more organized and form chromosomes, right? And to help condense themselves into sister chromatids, and chromatids are just 2 replicas of the same DNA, but they're just 2 different strands. Usually when you imagine a chromosome, you imagine a little X, right? Well, one side of this X has the exact same DNA as this other side, and they do this so that they can divide and one strand then gets pulled away. You will see this later, right? So here you can see our chromosomes becoming more organized right here, and then also the nucleus starts to dissolve. This little breaking line right here, that's the nucleus dissolving, so it can let the chromosomes get out of the nucleus and be ready to divide. Then the centrioles form spindle fibers. Now the centrioles are these structures right here, these little V's, and these structures are in the cytoskeleton of the cell, and they aid in forming the fibers. And you will see what the fibers are used for later. These are the spindle fibers right here, right? And so they form these spindle fibers, and centrioles move to the opposite side of the cells. You will see why in just a second. All right, so then comes metaphase, and you can remember this by M, and M stands for the middle, right? So why might that be? Well, the chromosomes line up in the middle of the cells, right over here. See, the chromosomes are in the middle of the cell. And the spindle fibers attach to the centromere. The centromere is this little circle right here, and it holds the sister chromatids together, right? This is one sister chromatid, this is the other sister chromatid, and the centromere holds them together. So the spindle fibers attach to what holds the sister chromosomes together. This is a bit of foreshadowing for what's to come, right? So this is all there is to metaphase. The cells line up in the middle, and the spindle fibers attach to the centromere of the sister chromatids. Then comes anaphase. Anaphase can be remembered as A. A stands for apart. And this is because the different sister chromatids get pulled apart by the spindle fibers to the opposite ends of the cell, right? This is what I meant with, you will see why chromosomes create a replica of themselves to be sister chromatids. Because you can imagine you have a chromosome like this, right? And each one has an identical set of DNA. That means that when they get pulled apart, one gets pulled over here, one gets pulled over here, one gets pulled over there. That means that that same DNA will be in both cells to come. So that's the objective of having 2 replicas of the cells and having sister chromatids, right? So the chromatids are pulled apart by the spindle fibers to opposite sides of cell, right? Then the last phase is telophase. And in telophase, the T stands for 2. And that is because now you will see two cells forming. This is not like cytokinesis, which is the last phase, but it's like pre-cytokinesis. You can imagine it like that. So what happens in telophase? So the cells start to come apart, and you can see this over here. They look more defined as two different cells than they did over here, for example. And the nuclear membrane starts to form. So this thing over here starts to form on both of the cells and separate them. And the nucleus starts to reform. So you can see this broken line. That's because the nucleus isn't fully reformed yet, but it's starting to get there, right? And the chromosomes are becoming more loose. So they're turning back into chromatins, which is the messy, mess that chromosomes are actually like when they're in the nucleus. So you can see there's just starting to become it. Like there's some organized ones over here, but there's some messy ones like this one over here on both sides. So this is what happens, and then comes cytokinesis, which can have a different video for itself. All right, so just to review a bit, remember the slogan PMAT: Prophase, Metaphase, Anaphase, Telophase. Prophase, the cell starts to prepare for division. Metaphase, the chromosomes line up in the middle. Anaphase, the chromatids get pulled apart. And telophase, the cell splits into two. So here are the diagrams once again. And I hope you now learned something new about the mitosis and the cycle, and I hope you remember this because it's really important and it's really fun to see how your own cells divide. And you can look under a microscope, take an onion, take a strawberry, look under a microscope of its tissue, and try to find each single one of these stages under the microscope. Alright, well, I hope you learned something and I hope you enjoyed this video, and see you next time.